You ever wondered if there's, more to us than just the physical, like maybe an aura, or even something like a soul that leaves when we die? You've probably seen stuff online, maybe pictures claiming to show some kind of glow around people, right?
Absolutely. It's a really ancient question, isn't it? Touches on science, philosophy, even spirituality. And today, we're doing a deep dive into the whole scientific quest to understand this human light.
Fantastic. Yeah. So our mission today is to really unpack the sources you sent over. We've got everything from those early kind of speculative ideas right up to the latest science.
Right.
We want to separate what's actually been proven from what's still more belief. We'll walk you through the research over the decades, uncover some really surprising things, and figure out what this light actually tells us about life. So where do we start?
Well, let's look at those first observations that kick things off. The sources show these images, like you said, of people where some parts are, normal blue, but then other bits are glowing green, yellow, red. It looks quite dramatic.
Yeah, it really does.
And the key thing here, this isn't just infrared body heat. You look at a thermal image, the heat's mostly in the torso, right?
Uh-huh.
But this glow, it shows up strongest around the face, for instance, in a living person. But then you see an image of someone deceased.
And it's gone.
Completely. The source image shows it as just blue and, lifeless.
And it wasn't just people, right? There was that rat comparison too.
Right.
Living rat, bit of a glow. Dead rat, nothing.
Exactly. And this observation, especially the difference between living and dead, led this brilliant Japanese scientist, Masaki Kobayashi, back in 2009, to ask this really fundamental question.
Okay.
He basically said, what is this glowing thing? It's there in living things, gone after death, and it's not body heat. What is it? And that question, it really launched this big work later on.
Okay, so Kobayashi in 2009, but you mentioned the story starts much earlier.
Oh, way earlier. We need to go back to 1923, Soviet Union. A Russian biologist named Alexander Gruvich.
Urich, okay.
His obsession was cell division. How do cells know when to multiply? That was his big question.
And he was looking at onion root tips under a microscope.
Yeah. And he saw something that just baffled her. He felt these cells were communicating somehow. Instantly. Invisibly. Telling each other to divide.
Instantly. Not through, slow chemical signals.
That was the standard thinking, yes. But he thought it was too slow. He expected something else. He came up with this hypothesis. Mitogenetic radiation.
Mitogenetic radiation, okay.
Invisible radiation, he thought. Signaling cells to multiply.
That's a pretty bold idea for the 1920s. How do you even test that?
With a really clever experiment, actually. He took one actively growing onion root tip, the sender, and aimed it right at another root tip, the receiver. But crucially, he put a thin piece of quartz glass between them.
Quartz glass, yeah.
To block any direct contact or chemical signals. Whatever got through had to be some kind of radiation, right?
Makes sense. And what happened?
Well, the results were pretty striking. After a few days, the cells on the receiver root, exactly where the sender was pointed, they multiplied significantly more than other areas.
Wow. So it worked.
He certainly thought so. He was probably influenced by Einstein's recent work on light, too, relativity from 1905. He was convinced this invisible light, this aura, existed and was signaling growth.
And he published this.
Yep, in 1923. Became a scientific sensation almost overnight. A real rock star.
But there's a twist, isn't there?
Always is, right? Yeah. Over the next few years, maybe 500 or so scientists tried to replicate his experiment.
500? That's a lot.
It is. And the results were, all over the place. Some saw the effect, lots saw nothing. And the really critical part, nobody could actually detect or prove the existence of the light itself.
Ah. So the core claim remained unproven.
Exactly. Critics like the physicist Irving Langmuir were pretty harsh. He called it pathological science, basically, wishful thinking leading to false results. But Gruvich, he remained 100% convinced, published 2 books on it just before he died in 1954. He essentially passed the torch to his daughter, Anna Gruvich.
So the idea persisted, but the proof of the light itself was still missing. Until, Anna, you said, in the 60s.
Right. Fast forward to 1962. Technology had moved on. Anna Gruvich got access to something called a photon counter multiplier.
Okay, what's that?
Basically, a super-sensitive device. It can detect and amplify extremely faint light individual photons, even.
So she tried to detect this light directly.
She did. Put plant cells in a totally dark box, aimed this detector at them, and bingo.
It detected light.
It detected photons, yes. Even in pitch blackness, that was the breakthrough. Living cells do produce light.
Well, why couldn't anyone see it or detect it easily before?
Two reasons. First, it's in the ultraviolet spectrum, UV light, around 200, 300 nanometers. Our eyes see visible light roughly 380, 780 nanometers, so it's invisible to us.
Okay, invisible. And the second reason?
It's incredibly faint. We're talking maybe 1 to 1,000 photons per square centimeter per second. Compare that to a regular 60-watt light bulb spitting out quintillions of photons. It's unbelievably weak.
Wow. Okay, so it's real, but it's UV and super, super faint. That explains why it was so hard to pin down.
Exactly. And this discovery, well, it kind of opened up a whole new perspective, a new dimension for looking at living systems.
So cells emit faint UV light. This must have reignited interest, right?
Oh, definitely. Which brings us to the 1970s, and a German scientist named Fritz Albert Popp. He was at the University of Marburg, got really fascinated by Gorvich's work, especially thinking about cancer research.
Popp. Okay, and he gave this light a name.
Yeah, he coined the terms we often hear now, biophotons or ultra-weak photon emission.
Photons, right.
But he went much further than just naming it. His big claim was that these biophotons weren't just like random waste light. He proposed they were actually a sophisticated communication system.
Communication, like Gervich thought.
Kind of, but much more complex. Popp theorized this light regulated basically everything in our cells and bodies: DNA repair, growth, maintenance, the works.
How would that even work with such faint light?
Well, he brought in this concept from quantum mechanics, coherence. He suggested the biophotons weren't emitted randomly like a normal light bulb that's incoherent light, messy.
Okay. He thought they were emitted in ordered, focused patterns, like laser light.
Coherent. That way, he argued, even faint light could carry complex information efficiently within the body.
Coherent biophotons. Like a biological laser network. What was his evidence for that?
It was mainly statistical analysis of the photon emissions. He claimed the data showed photons being emitted in fixed patterns, not randomly, like synchronized music was the analogy he used.
And this led to a bigger theory.
Yeah, his grand theory was that biophotons are like a cellular radio. Emitted, he thought, by the DNA itself, broadcasting signals for growth, repair, immune response, everything.
Wow. And he connected this to disease.
He did. He suggested that glitches in these biophoton signals could lead to illness. He even proposed that cancer cells have disturbed coherent patterns, meaning you could potentially detect cancer really early just by studying a cell's light emission.
It's a huge claim. Early cancer detection through light patterns. Absolutely. He published all this in his 1989 book, Biophotonics. He was apparently very charismatic, very inspirational, founded the International Institute of Biophysics in Germany. He's often called the father of biophotonics.
Okay, so Popp's ideas sound revolutionary, but also maybe a bit out there. How did the scientific community react?
Well, this is where things get really interesting and honestly a bit messy. Popp's ideas basically split the scientific community right down the middle.
Two camps.
Pretty much. Yeah. You had the believer scientists who were inspired, saw huge potential, and started researching biophotons based on his framework.
And the other camp.
The disbelievers, skeptics who thought his interpretations, especially the coherence part and the DNA communication idea, were way too speculative, lacked solid proof, and they started looking for alternative, more conventional explanations for the light.
So, a real divide. And this is where science started to blur with other ideas.
Exactly. This period saw the lines get incredibly blurry for decades. People started taking the idea of body light and running with it in directions Popp himself might not have fully endorsed.
Like who?
Well, take Konstantin Korotkov, a Russian scientist in the 1990s. He was inspired by Popp.
Yeah.
His claim was pretty mind-blowing. He said that after death, the body gradually releases its consciousness, like an aura, over 24 to 48 hours.
Releases consciousness as an aura. How would you even measure that?
He developed a device called gas discharge visualization, or GDV. He claimed it could measure a person's biofield, this invisible energy field he believed reflected physical, mental, and spiritual health.
A biofield. That sounds spiritual.
It leaned heavily that way. His 1998 book, Light After Life, made really controversial claims, like the dying body releases this aura consciousness, measurable by GDB. He even said the way someone died affected the aura.
How so?
Like a violent death supposedly resulted in an intense, irregular, long-lasting aura reflecting trauma. A peaceful death, a smooth, steady release.
Those are extraordinary claims. Did the GDV device hold up scientifically?
Not really, no. The mainstream scientific view was, and still is, pretty critical. Many analyses concluded the GDV device was basically just measuring changes in skin moisture and conductance, things that naturally change with temperature, sweat, stress, not some mystical biofield. Reproducibility was a major issue too.
Okay, so Kurtkov's claims were largely dismissed by mainstream science. Who else was blurring these lines?
Then you have Masaru Emoto, the Japanese author, maybe scientist isn't the right word, from the 1990s. His work became incredibly popular, still is for some.
Emoto, the water crystal guy.
That's the one. He took this light consciousness idea even further, said consciousness is universal, even affects water.
Right, the experiments where he exposed water to word.
Exactly. His famous setup: distilled water, exposed to positive words, love, gratitude, peace, versus negative words, hate, evil, stupid. Then he'd freeze the water samples.
And the claim was the positive water made beautiful crystals.
Yep. Supposedly formed healthy, structured, symmetrical ice crystals. The negative water, asymmetrical, unhealthy, irregular structures. He even claimed tap water had negative energy compared to pristine mountain water.
His book, The Hidden Messages in Water, was a massive bestseller, wasn't it?
Huge. New York Times bestseller. Still highly rated online today. People really resonated with the idea.
But scientifically.
Widely considered pseudoscience, lack of controls, no blinding, inability for independent researchers to reproduce the results consistently. The results seemed highly subjective.
So this really raises that question again. Was science actually connecting to spirituality here? Or were these popular ideas maybe just wishful thinking riding the wave of the biophoton discovery?
That's precisely the tension. Which brings us back to that other school of thought, the disbelievers, the skeptics.
Right, the scientists who weren't buying Pop's grand theories, including Masaki Kobayashi, the guy who asked the initial question.
Exactly. Their view was, look, Pop's ideas about coherent communication from DNA are interesting, but they're speculative. There's not enough hard evidence. There must be a simpler, more biological explanation for this faint light.
And this wasn't a totally new line of thinking either, was it? Didn't someone suspect a chemical cause way back?
You're right. Alexander Hollander, back in the 1930s, had already hypothesized this light might just be a chemical byproduct, like bioluminescence in fireflies, but much weaker. He just lacked the tech to prove it then.
Oh, okay.
Then, in the 1950s, two American scientists, Bernard Streller and William Arnold, used those early photomultipliers like Anna Gurvich later used.
What did they find?
They did rigorous tests, mainly on plants and algae. And crucially, they published their findings in a proper peer-reviewed journal, the Journal of General Physiology.
And their conclusion?
They found the light emission was directly proportional to the plant's metabolic activity, things like photosynthesis. They linked it directly to chemiluminescence.
Chemiluminescence, light from chemical reactions.
Exactly. So the idea that this was linked to basic metabolism, to chemical reactions, was already there in the peer-reviewed literature decades before Pop's more elaborate theories took off.
Which brings us full circle, back to Masaki Kobayashi. The biggest skeptic, you said, but also the one who connected the dots.
Yes. Kobayashi was brilliant. He respected the early work, acknowledged the light existed, but he was deeply skeptical of the communication theories. His driving thought was, we need to find the root cause of this light. And to do that, he realized, needed much better technology.
The technology gap again. What did he use?
This was his big innovation. Around 1999, working with the company Hamamatsu Photonics, they developed a super advanced camera, a cooled, charge-coupled device, or CCD camera.
Cooled by cooled.
Cooled down to minus 120 degrees Celsius. This almost completely eliminated thermal noise, random signals generated by heat, which allowed the camera to be unbelievably sensitive.
Sensitive.
We're talking literal single photon detection. It could pinpoint the exact location and time each photon hit the sensor. It could create real-time 2D maps of how photons were flowing. Plus, it had high-throughput lenses to capture as much of that faint light as possible. It was a generational leap.
Okay, so armed with this incredible camera, what did he do?
He conducted what I think is a truly historical experiment using a rat's brain.
A rat brain. Okay.
Yeah, expose the brains, put them in a totally dark room with the CCD camera running, but simultaneously he was measuring the brain's electrical activity using an EEG.
Electroencephalogram, right?
Right. And also measuring blood flow and oxygen levels, CBF, cerebral blood flow. So he had brain activity, metabolism indicators, and the light emission all measured at the same time.
And what did he see when he put all that data together?
Really interesting patterns. Whenever the brain's electrical activity spiked, or when oxygen levels surged, both indicating neurons firing and metabolism ramping up, Guess what happened?
The brain emitted more photons.
Exactly. More brain activity, more metabolism, more light.
Okay, so the light is directly linked to metabolic activity.
Precisely. And what's a key byproduct of metabolism, especially when cells are stressed or working hard?
Waste products.
Specifically, reactive oxygen species, ROS.
ROS, right, those unstable oxygen molecules.
That was the crucial link Kobayashi made. His big discoveries, backed by this solid evidence, were: 1, a rat's brain literally glows more when it's more active or stressed. The light is from cell metabolism.
Okay.
2, metabolic stress increases ROS levels. And 3, as ROS levels go up, photon emission goes up.
So ROS looked like the hidden culprit behind the light.
It strongly suggested it, but he needed definitive proof, which led to his really smart Kauki plant experiment in 2007.
Kelpie plants. Why them?
Because he could deliberately stress them in a controlled way. He sprayed them with a virus cucumber mosaic virus, which triggers an allergic inflammatory response in the plant.
And that response increases metabolic stress in ROS.
You got it. Then he did something clever. He took one of the infected plants and injected it with a powerful antioxidant called Tyrin.
Antioxidants.
Yeah, they
neutralize ROS, right?
Exactly. Tyrin specifically destroys ROS. So he had stressed plants, some treated with an ROS killer, some not. And the camera was watching.
And the results.
Undeniable, crystal clear. First, exactly where the plant was fighting the virus infection, the photon count shot way up. The stressed parts glowed much brighter.
Okay, confirms the link to stress metabolism.
But second, and this was the clincher, in the plant injected with the antioxidant Tyrin, the photon intensity dropped dramatically by 50 to 80%.
Wow, so removing the ROS removed most of the light?
Precisely. That was the smoking gun. The clear conclusion: reactive oxygen species are the main cause of these biophotons.
That's huge. It completely changes the narrative from cellular communication to, chemical reaction.
It really brought the whole field back down to earth, back into the realm of practical, evidence-based biochemistry and cell biology, away from the more speculative interpretations.
Okay, so ROS causes the photons. But how? What's the actual mechanism?
Right, let's unpack that. It was clarified further in reviews around 2014. Basically, think of ROS as unavoidable waste from making energy in our cells, in the mitochondria mostly.
Okay, waste products.
And these oxygen-based molecules are reactive because they're chemically unstable. They're either missing an electron or have weak bonds, so they desperately want to react with other molecules.
They want to steal electrons.
Pretty much. They try to snatch electrons from important stuff nearby, like DNA, proteins, fats, and cell membranes. This electron stealing damages those molecules.
Right, that's oxidative stress.
Exactly. But here's the key physics bit. When these electrons get shuffled around, transferred from one molecule to ROS, energy is released. That electron transfer reaction
Releases energy as light.
releases a tiny bit of energy in the form of a photon. It's the chemical reaction itself, the electrons shifting energy states, that generates the light particle. That's chemiluminescence happening inside our cells.
So our body glows because of the constant chemical reactions involving these reactive oxygen species.
That's the core scientific understanding now, yes. It's a byproduct of being alive and metabolizing.
Okay, so this means we probably can't detect a soul or consciousness leaving the body with this light, like Kowalkoff suggested.
Highly unlikely based on this evidence, yes. The light seems tied directly to ongoing metabolic processes, which cease after death.
But, and this feels like a big but, if the light is directly linked to metabolism and stress, especially ROS levels, doesn't that open up other, maybe more practical possibilities?
Absolutely. This is where it gets really interesting from a medical perspective. Forget the soul for a moment. We can potentially use this light to detect deadly diseases like cancer at a very early stage.
Because cancer cells have different metabolism.
Exactly. They often have highly abnormal, ramped-up metabolism and produce more ROS as they grow and divide uncontrollably.
And Kobayashi showed this.
He did. Even back in 1995, using mice, he found cancer cells glowed 1.5 to 4.7 times more intensely than normal cells.
Wow.
And by 2004, studies confirmed this in humans. 2 advanced cancer cells emit significantly more photons.
The potential there is just immense. Detecting cancer before a tumor is even visible on a scan just by seeing abnormal light patterns from cells.
It could be revolutionary. Early detection is so critical for cancer survival rates. This could genuinely save countless lives.
Are there other applications emerging besides cancer?
Oh, yes. It's a whole field now. For example, back in 2009, Kobayashi's lab showed our overall body glow follows our circadian rhythm.
Our body clock.
Yeah. We tend to glow slightly more during the day when our metabolism is higher and less at night when we're resting. It reflects our internal clock.
Cool.
In 2019, there was this beautiful study imaging the changing glow patterns as a caterpillar transforms into a butterfly, mapping the metabolic shifts during metamorphosis.
Amazing.
Also in 2019, researchers showed they could detect UV-induced skin damage, like from sun exposure. UV damage increases ROS in skin cells.
So the skin glows more afterwards.
Precisely. You can literally see the stressed areas lighting up.
The medical potential seems almost limitless then, detecting inflammation, allergies, maybe even genetic mutations based on their metabolic glow signature.
That's the direction things are heading.
Yeah.
Imagine combining this ultra-sensitive imaging with AI that has learned to recognize the photon patterns associated with different conditions. Doctors could potentially spot problems incredibly early, non-invasively.
That's really exciting. But it does raise a question for me. We've been talking about ROS as this damaging thing, the source of this light signal for problems like cancer, UV damage. Is ROS always bad?
That's a fantastic question. And the answer is crucial. No, ROS is not always bad. There's nuance here.
Oh, okay.
In normal, controlled amounts, ROS is actually essential. Our body uses it deliberately as an important signaling molecule.
Signaling how?
It helps trigger processes like recovery, detoxification, immune responses. It tells cells what needs doing.
Can you give an example?
Sure.
Muscle building after a tough workout, that stress generates some ROS, right?
Yeah.
Well, that ROS actually acts as a signal to tell the muscle cells to ramp up protein synthesis and repair, to get stronger. It communicates the need to adapt.
So the stress signal is actually useful there.
Exactly. In fact, some studies have shown that taking high doses of antioxidant supplements like vitamin C or E right before or after exercise can actually blunt muscle growth and adaptation. You're mopping up the signal the body needs.
Huh. So it's about balance. Too much ROS is damaging oxidative stress, but a certain amount is necessary signaling.
Precisely. Which, in a funny way, brings us back to Gurvich.
How so?
Well, he was wrong about the mechanism. It wasn't coherent light messages. But he was kind of right in principle, wasn't he? He sensed there was some kind of invisible signal involved in growth and function.
And the underlying reality ROS signaling does perform some of those roles.
Exactly. It's just biochemical signaling, not light signaling. It shows how science progresses, right? An initial insight, maybe misinterpreted, points towards a deeper truth that later technology and experiments uncover. It's a beautiful journey.
It really is. We've gone from these ancient questions about souls and auras.
Right. Through those early onion root experiments, the debates, the mixed results.
And the more speculative theories of Pop, Korotkov, Emoto, blurring science and spirituality.
Before finally landing on this precise, evidence-based understanding driven by technology like Kobayashi's camera.
The ROS connection.
Yep. The chemiluminescence linked directly to metabolism.
And it really underscores the importance of, well, skepticism and rigorous proof in science, doesn't it?
Absolutely. It highlights that huge difference between compelling stories people want to believe, like GDV measuring auras when it's likely just skin moisture, or water responding to words, and the hard-won, peer-reviewed evidence that stands up to scrutiny.
Pop's work was clearly foundational in focusing attention on biophotons.
Definitely. He coined the term, spurred research. But some of his specific interpretations, especially about coherence and direct communication, just didn't hold up under further investigation, which is normal in science. Ideas evolve as evidence grows.
So this whole deep dive, it not only reveals this amazing fact that our bodies literally glow chemically, but it's also a powerful lesson in telling the difference between solid scientific proof and maybe sensational speculation. That just sounds good.
Well said.
It makes you wonder what other phenomena that seem mystical today might just be waiting for the right technology or the right experiment to reveal a fascinating but ultimately scientific explanation. What really stands out to you when you look back at this whole journey from aura to ROS?